Okay, now we'll move on to our second nucleic acid, RNA. RNA stands for ribonucleic acid. So just like DNA, the ribo stands for the sugar, ribose sugar, and of course they're both nucleic acids. Now you can see RNA here. It is a single-stranded structure, different from a DNA. It is not in the shape of a letter. It is comprised of nucleotides, which again are made of phosphates, sugars, and a nitrogen base. The sugar is ribose and it has one extra oxygen compared to deoxyribose. And then the bases are going to be slightly different. There's A, U, C, and G. I have another memory device to help you with these. All unicorns, so that's adenine with uracil, because there is not a thymine, can gallop. So all unicorns, A with U, can gallop, C with G. And here's another one that I really like too. Always remember, RNA loves you. Okay, now the process of making RNA, which we just saw in our little video there, that RNA is the workhorse. RNA is what's going to be taking the genetic code from the DNA that resides in the nucleus out of the nucleus and to ribosomes for assembly into protein. So RNA is the go-between in what Francis Crick termed the central dogma, but today we just refer to it as gene expression. So the DNA, the double helix, has all of your genetic code for all of your proteins, for all of your characteristics, but it's too big to leave the nucleus, but it can pass that information along to the RNA, which is single-stranded. It can leave the nucleus and then travel to a ribosome. Okay, so let's do this transcribing or this rewriting of the genetic code into RNA using the RNA alphabet, which is A, U, C, G. Those are the only four letters that you have. So we're just going to take one strand of DNA here because this is the way it would work. Let's say that you hurt yourself and you need some more muscle fibers, then signals are going to be sent to the nucleus to switch on the gene for that protein that you need. Just that gene for just that protein, not all of them. So here's our DNA, and let's go for it. C will go with G, G will go with C, T will go with A will go with U. So there isn't a T. So the A's will go with U, the G with C, C with G, G with C, G with C, C with G, and then T with A. So A, U, C, G are the only four letters in the RNA alphabet. So you really just have to be careful when you have an A over here. Now at this point, the new single-stranded RNA structure is made. It will detach from this DNA. It's not going to stay with the DNA with hydrogen bonds like we saw when we replicated DNA. It's going to detach and then it's going to travel to a ribosome to take this code that it just transcribed, it just rewrote this code from the DNA into its own alphabet, and it's going to take this code out to a ribosome to translate it into whatever protein that you need, which might be something to replace a muscle fiber. So here's one for you to practice on your own. So I'm going to give you a few seconds to kind of think about this, and that all unicorns can gallop. Okay, so this is what you should have. G with C, A with U, because there isn't a T, G with C, T with A, T with A, C with G, C with G, G with C, A with U, T with A with U, and C with G. And then again, this piece of RNA is going to leave and go do what it needs to do. Whoopsie, that must be a mutation. Okay, there are three types of RNA involved in the next process that we'll get into, and we'll just talk about it briefly later on. Translation, which is taking that genetic code and translating it into one of your proteins. So the three types are mRNA, tRNA, and rRNA. Now, the M, the T, and the rRNA, they are shortened. M is for messenger, T is for transfer, R is for ribosomal. So we'll start with the ribosomal RNA. The ribosomal RNA actually makes up the ribosomes themselves. Then we'll go to the messenger. The messenger RNA brings the message, and in this case, what the message is that genetic code from the DNA. And then the T is for transfer, but I like to say transfer, transport, or take a friend. So the transfer RNA is actually going to bring amino acids, which are the little monomers or the building blocks of proteins, over to the message and put them together according to the message from the DNA. So when you think about this, I like to think about construction, because that's what's happening, construction of proteins. And that process is going to be called translation. So the messenger RNA kind of acts like the blueprint. And this blueprint comes from the DNA by way of the RNA. So remember, DNA has all the information, but it's ginormous. It can't leave the nucleus. But RNA is a little guy, it can. So the messenger is going to bring out this message to the ribosomal RNA, which is the construction site. And then the transfer RNAs, these little guys, are going to read this message and bring the amino acids over and put them in the order that they go. So translation is what I just described, which is the process of making protein, and we will come back to it later.